Hello, and welcome to our lecture on DNA tech, or as the long way to put it, modern applications of microbial genetics. What we learned in microbial genetics, we can apply to some rules in the laboratory. And if you use those tools and select ways and highly manipulate that, you become a molecular biologist because you are manipulating the bacterium on a level of which it is not natural, right? So you're trying to maybe make a bunch of insulin or another type of gene in a type of plant to see what it does. So in this lecture, we're going to discuss the application of genetic engineering, like I was just talking about. What can we do with genetic engineering? Define transformation of recipient cells and discuss how it pertains to antibiotic resistance plasmids, and then we can discuss polymerase chain reaction, or PCR. And all of these are kind of interrelated, and this should be a nice, short, sweet lecture for us. Since we can't really get into most of this technology, we will just kind of discuss it here. All right, so one of the main tools used in molecular biology, or our applications of microbial genetics, is the thermocycler here. Okay, it's a machine that can hold a small piece of plastic with 96 holes in it that are contained. They are called wells. 96 wells can go into the machine. Some machines are larger, and it can have more plates. So it can have like 4, maybe 8 plates. But all those plates are 96 individual wells, so potentially 96 individual samples. You would include controls in there, and controls are a known positive or a known negative. And especially if you're going to be testing someone for chlamydia or C. trachomitis. Okay, if you're testing for someone with chlamydia, you are going to need to use the thermocycler to do a DNA test and see if a unique sequence of chlamydia is within your bloodstream. And if it is, you will probably likely be treated for chlamydia. Okay, a few more tests would be done. But interesting to note, the most common cause of blindness in the world is chlamydia. Okay, and it is the most common sexually transmitted disease. So when we learn about chlamydia in the future, we kind of already touched on it in the first unit, but when we touch on it again, you'll know that C. trachomitis is one of the most commonly sexually transmitted disease. The most. Okay, and we need to use this thermocycler to do it. Okay, and we'll get into how the thermocycler can test that for us. Okay, first we need to describe what recombinant DNA is. Okay, we are going to take a piece of that DNA from a gene in a human cell that produces insulin, right? So maybe a pancreas cell or even just any kind of human cell will turn on that gene. We'll take that insulin gene and we'll put it into a plasmid. We'll cut open this plasmid and insert that gene there. Okay, as you see here, and it becomes a recombinant DNA plasmid. Okay, then we put that into a bacterium that is known to not produce insulin, or something that infects another organism, like we do with plants. Then the end result is still the same, is to take that gene. Now that organism produces that gene, that protein. Okay, and the idea is that it is recombinant DNA. Okay, and now we can create insulin on a mass level. So how did we cut it open? Well, we cut it open using a plasmid vector. Okay, we have all these sites right here, the MCS, the multiple cut site, and it's called a multi-cloning site, but easy to remember that it's a multi-cut site. We can cut it there using specific enzymes, those EcoRI, XBA1, PST1, Hindi3. We can use those to cut specific sequences, and then we add specific sequences onto that insulin gene so we can glue it in. And we use that DNA ligase that we learned in DNA replication, right? We use that DNA ligase protein to ligate these together, okay? And we can include a reporter gene, like that's fluorescence. When we shine a light on it, it fluoresces. That was in Sherlock Holmes. There was like a fluorescent bunny rabbit running around. There's fluorescent mice. The thing is, you need to shine a UV light on it to see it. You also have a blue-white screening method that you can also see, and it's either blue protein made or not. And that tells you that you got your gene in. Okay, so molecular cloning takes advantage of the terms we already learned: transformation, conjugation, and transduction. Okay, so that's kind of what we want to take away from this is remember, transformation is taking the DNA from a foreign entity, just from the environment, okay, and we force it to do that with plasmids. We take advantage of the technology of a plasmid when a plasmid goes through the conjugation, and now that plasmid helps that bacterium have an antibiotic resistance, say. And then transduction also is what we need, because we need to actually incorporate into a gene just like a virus would, and sometimes we take advantage of viruses to do this. Okay. In plants, as I mentioned, we use an intermediary called agrobacterium. Okay. Agrobacterium is what we use to actually infect the plants, because that's what the bacteria wants to do anyway. It incorporates its DNA into the plant and creates a tumor. Well, if you take out the tumor producing genes and you then make a gene of choice that you want in it, and you can test all these sorts of genes and say, if I take out this gene, what happens? If I take out this gene, what happens? Most of the time you kill the plant, but sometimes you get some cool phenotypes, some cool looking ways you affected it with this extra gene or with a knockdown gene, one that doesn't work. Alright, so in the homework it goes into polymerase chain reaction here. Alright, and basically what we want to do is we want to denature our protein. Okay, we're going to denature them. They're going to come apart. Our primer that we add in there, just like we needed an RNA primer, we're going to add little bits of DNA primer. We order those, we mix them up, put them in this reaction, okay, and then we put it in the thermocycler. Those primers are on either side, and they both go, allow it to go in the way that it needs to go, right, so that it can duplicate, okay, and then we will add the polymerase will be set to its optimal temperature. The polymerase will find those primers, and those primers will elongate or extend the nucleotides, okay, and then eventually you have 2 copies of DNA at the end of it, okay. So we went from denaturation, we took them apart, we annealed our primers to the templates, then we extended them or elongated them, and at the end of that we had 2 full different copies, okay. And then just like in binary fission, where we wait 20 minutes for a duplication, here we can wait literally a minute, minute and a half, sometimes even 50 seconds for one cycle, and it continues, okay, and then we just amplify it and we get millions and millions and millions of copies, okay. So cycle 2, we went from 1 copy to 2, 2 to 4, 4 to 8, 8 to 16, and we keep it going. And those primers, we add a lot of primers, so there's so many primers. We add a lot of polymerase, so the polymerase can work. Yeah, so the polymerase is the most expensive thing, and that's basically our limiting reagent, and why we usually go for like 30 cycles, because we only add the optimal amount of polymerase. Okay. When you have finally your PCR, you then will run it on a gel here, as you see, and that is how you can visualize the DNA that you amplified. Otherwise, you just have DNA in a tube. Can't really do much with that. And so this is how you would see, okay, I don't have this segment of gene and these two organisms, right? It's missing from here. This one doesn't, all of these don't have this. And so those are mutants of some kind compared to these, right? All right, and so we'll get into a little bit of gene therapy in the next video, and they will end.